My dear students, welcome to my today's online lecture class, continuation of topics of recombinant DNA technology. We have learned so far overall view of what happens in recombinant DNA technology experiments. Then you have learnt about molecular tools, that is restriction endonucleases and DNA ligases and alkaline phosphatase. Knowing about enzymes is one requirement to conduct recombinant DNA experiments, but apart from enzymes, you have to know so many other factors which are needed for the successful completion of an RDT experiment. So, I am Dr. Meera BK, Associate Professor and Head, Department of Zoology, Maharani Science College for Women, Palace Road, Bangalore 1. You can see my cell phone number and email ID on the slide. After the talk, if you have any queries, any questions, do contact me either over my cell phone or to my email ID. Now, let us know about vectors, another important tool that is needed to conduct recombinant DNA technology after learning ourselves about the various types of enzymes that are needed. Now, what is a vector? Vector is basically a Latin word. The etymology goes like this: vihir. Vihir means to carry. So vector means a carrier. You must have studied as zoology students vectors in parasitology, but there vectors is altogether a different aspect. In recombinant DNA technology, though they are used in the same sense, that is as carriers, their role to be played is totally different. What is the definition then? Here in RDT, a vector is a DNA molecule that has the ability to replicate in an appropriate host cell and into which the DNA fragment to be cloned is integrated for cloning. Here you should note two things. One is a vector should have the ability to replicate in an appropriate host, whether it is a bacterial cell or a plant cell or an animal cell. And one more thing is it should be able to successfully integrate the DNA fragment of our choice in which we are interested to bring about gene cloning. So, a vector is a DNA molecule that has the ability to replicate in an appropriate host cell and into which the DNA fragment to be cloned, what we call DNA insert, is integrated for cloning. Now, please note, vectors as such do not exist in nature. We have to design our own suitable vector for a particular experiment in the laboratory, picking up so many components from different sources. Okay? So, please note, none of the vectors exist naturally. Okay? They are all designed so as to suit to our purpose of experiment. Now, what are the characters of an ideal vector? If I want to have a vector, what are all the characters I expect to be present in it? One is they should be capable of autonomous replication. They should be able to replicate on themselves. When it is possible, only when they have their own ori of replication, origin of replication. So this topic needs a little bit background of molecular biology, Hope you have studied in your previous semesters. So first thing, it should be capable of autonomous replication. That means a vector should have its own ORI of replication. Then it should be easy to isolate and purify. It should be easy to isolate and purify. This is one more character what I expect in vectors. Then a good vector should enable easy transformation. A good vector should enable easy transformation. What do I mean by transformation here? Transformation is a process of gene transfer by which some bacteria take up foreign genetic material. I repeat, it is not there on the slide. Please note down. Transformation is a process of gene transfer by which some bacteria take up foreign genetic material. Okay. So a good vector should enable easy transformation. It should get transformed easily. Then should have suitable marker genes. Should have suitable marker genes so that screening and selection of the transformed host cells will be easy. See, screening and selection of the recombinant host cells is altogether a big topic in RDT, altogether a different chapter where there are different methods, and unfortunately it is not covered in your syllabus. But right now you just know they should have a suitable marker genes so that selection and screening is easy. What do I exactly mean by a suitable marker gene? A suitable selectable marker is a gene introduced into a cell, especially a bacterium or the cells in culture, that confers a trait suitable for artificial selection. That confers a trait for artificial selection. There are different methods like insertional inactivation, alpha complementation, immunoprecipitation, immunological methods, etc. But you are not studying that aspect in detail in this paper. Okay? Now, should contain unique target sites for as many as restriction enzymes as possible. Should contain unique target sites for as many as REs as possible. So here, restriction enzymes I have taught you already, and by now you must be knowing what are target sites. So these are all the properties, good properties that I expect to be present in an ideal vector. Now, after knowing the properties of a good vector, you should know the types of vectors. Normally, we study two types of vectors in RDT. One is cloning vector, another one is expression vector. What are they? Let me tell you. One is cloning vector in the sense, these are small DNA vehicles that carry a foreign DNA fragment. Foreign DNA fragment. Okay. And here the purpose of designing such a vector is just to propagate the DNA of your choice. Like you want to have multiple copies of the DNA insert. Okay. In such cases you will use cloning vectors. So the main purpose is propagation. Once you get multiple copies, you may use them for different purposes like research, for further studies and other things, right? Here expression is not the motto. The retrieval of proteins is not the motto. So it is a small DNA vehicle that carries a foreign DNA fragment. Best example for this are plasmids as vectors and lambdaphages. That is a type of bacteriophage, right? Now, we are studying mainly cloning vectors as per your syllabus. Then the second type is expression vectors. It is a relatively a small DNA molecule used to carry a specific gene into a target cell. Used to carry a specific gene into a target cell. Inside the host cell, the protein that is coded for by the gene is produced. So here the motto is to produce the proteins that is coded by the DNA of your choice. Okay, by regular transcription and translation processes of the host cell. So in cloning, getting multiple copies is the motto. In expression vectors, retrieving the required protein is the motto. So there are two types of vectors which are normally used in RDT. Okay, but we are studying only about cloning vectors. All right. Hope it's clear. Then, depending upon the host, vectors are cloned into which the vectors are cloned. Okay. The expression vectors are classified into two types. Say for example, if you put an expression vector into a prokaryotic cell host, we call them as prokaryotic vectors. On the other hand, if you put them in an eukaryotic cell, host cell, we call them as eukaryotic vectors. Okay. So after knowing about the definition, properties of a good vector and the types of vectors that which we normally use in RDT, now we should study different types of vectors wherein according to your syllabus you are studying only three. One is plasmid vectors, another one is bacteriophage vectors, another one is cosmids. Okay. Now in this class we will study about plasmid vectors. Rest of the two we will be studying in our next class. Now coming to the plasmid vectors. What are they? Before going into the vectors as such, you should know what are plasmids. Plasmids are extrachromosomal DNA elements capable of independent replication and propagation. The whole genetic unit containing the area of replication and its associated control elements is called a replicon. So another teacher will be teaching you about recon, muton, etc. in another genetics class, but right now we just know what is a replicon. So plasmids are extrachromosomal DNA elements capable of independent replication and propagation, and that entire unit containing the area of replication and relevant associated control elements is called a replicon. Now, after knowing the properties of an ideal vector in general, now you should know the properties of a good plasmid vector. Number one, smallest possible so that it is easy for isolation and handling. That's the only purpose why you prefer smaller ones. Then they should be capable of autonomous replication. Okay. Then they should contain single restriction site for as many as restriction enzymes possible. Okay. Then they should have clear selection markers. Just now I have taught you this to confer a selective phenotype on transformed cells. I will tell you a little later what exactly this is. Then restriction site should be available at least in one of the marker genes. When I take up the example of PBR322, I will explain what exactly I meant. Then for biosafety purposes, please note they should not be conjugative. They should not be conjugative because sometimes, if they are conjugate and by chance, if they are released into the environment, it becomes very hostile. So for biosafety purposes, they should not be conjugative. Now, one best example for plasmid vector is PBR322, with which all of you are familiar. Now, PBR322, what it stands for? P, it stands for plasmid. It tells you that it's a plasmid vector. BR stands for the people who developed it, Bolivar and Rodriguez, who developed the plasmid, and 322 stands for plasmid serial number. Okay, then yeah, this is the picture of the PBR 322, okay, which is an Escherichia coli cloning vector. That means you can clone into this E. coli cell. Host cell is the Escherichia coli. Now let us see the properties of this PBR 322. It is 4.3 kb Daltons. It contains 4.3 kb base pairs. Okay. Then 4.3 kb base pairs. Then it contains ORI, origin of replication, which is derived from PMB1, which is derived from PMB1. So the ORI is derived from PMB1. Then second one, it has tetracycline resistance gene. You can make out here in the diagram tetracycline resistant gene, the almost pink colored structure, tetracycline resistance gene. This is derived from PSC 101. Students, I have already told you none of the vectors exist naturally. You have to pick up different elements from different sources and design the vector of your choice. So ORI is derived from PMB1. Then tetracycline resistance is derived from PSC 101. Then ampicillin resistance gene, which is there right opposite to tetracycline resistant gene, that is derived from TN3. TN3 is a part of R1 DRD90. Okay, then it is one of the standard cloning vectors and is the grandparent of many other vectors. So many other plasmid vectors have been derived from this PBR 322. So you can see it has molecular weight of 2.6 Daltons and it has 4.3 kbp, and then you can make out in the tetracycline resistance gene region there is a site for BAMHI, SPH1, SAL1. On the other hand, in ampicillin resistance gene there is a site for SCA1, PST1, and then next to that you have SSP1 region, HIND3 region, in between you have EcoR1 target site region, and then below tetracycline resistance gene you have PUV site, and then adjacent to that you have ORI of replication. So this is how PBR 322 looks like. Then the advantage of this vector is there are over 40 enzymes. There are over 40 enzymes with unique cleavage sites. 40 enzymes with unique cleavage sites, and out of 40, 11 of them are in tetracycline resistance gene and 6 of them are in ampicillin resistant gene. Okay? Hence, cloning, when if you want to clone the DNA of your choice, it can be done using these 17 enzymes as they result in insertional inactivation of one of the marker genes. Okay? Either the ampicillin resistance gene or the tetracycline resistance gene. Okay? This can be used to select the transformed cells. Now, I have used the term insertional inactivation. It is one of the methods normally used for screening and selection of the recombinant colonies. What exactly I mean? Note down, it is not there in the slide. Insertional inactivation is a technique used in RDT. The bacteria carrying recombinant plasmids or a fragment of foreign DNA is made to insert into a restriction site inside a gene to resist antibiotics, thus causing the gene to turn non-functional or to turn into an inactivated state. Say for example, see here you can see the tetracycline resistant gene, right? If you have a DNA of your choice, and if you cut here by making use of BAM-HI and insert the DNA of your choice, the DNA insert will sit in between the tetracycline resistant gene. When it happens, this tetracycline resistant gene becomes inactive. It becomes inactive. By inserting the DNA of your choice, you are making tetracycline resistant gene inactive so that it will be susceptible to that particular antibiotic. So that screening and selection will be very easy, right? So selection of the transformants is by insertional inactivation, and that is followed by replica plating method. Okay. So children, so far I have taught you about vectors, properties of a vector, types of vectors, and one particular type of vector, that is plasmid vector, with one example. Thank you for your patient hearing. Stay blessed. Thank you.